I look forward to tell you a bit about the work that we do in our lab, really in trying to bring systems biology and several modeling framework to study the microbiome. And specifically what I'll talk about today is about several methods and research paradigm that we develop to use models, both metabolic models and mathematical models, not only to represent or simulate the microbiome, but actually to analyze microbiome data and specifically to integrate microbiome data across multi-omics. And so before I can do that, I feel that I have to start with a few clarifications. And so the first one is that I'm actually not studying HIV directly in any way, shape, or form. And so many of the faces in this room are new to me, and I'm hoping to get to know you and work with you. But the method that I will describe are a general method that we developed that should work for any kind of microbiome data, and I'm hoping that some of them would be useful to folks in this room. The second clarification, which I think is probably more important, is that I flew yesterday from Seattle, so as far as I'm concerned, it's a bit before 7 a.m. The sun has not yet risen in Seattle, and as a computational biologist, I'm not a morning person in any way, and so I hope you will forgive me if I'm not as sharp as I would like to be. And the last clarification has to do with the title, which I think is pretty descriptive in terms of the thing that we do in the lab. But within this talk, I really think that I want to focus on one take-home message, which is much simpler than that, and it's simply beyond associations. And that's what I'll try to convince you today, that as a field, we really have to move beyond making lists of genes and species and looking for correlation, which I think is a great first step, but it is limited in the sense of allowing us to understand the microbiome as a complex system. And so I want to really start with a quick motivation for why we need to do that. And as the first talk of the day, I thought it makes sense to start with the very first microbiome research, which, as many of you know, has been done by this guy over here, Antoni van Leeuwenhoek. He's considered by many the father of microbiology, a very odd fellow in many different ways. And back in 1683, he wrote this beautiful letter to the Royal Society of London. I will not have time to read the entire letter. If you are not familiar with that, I strongly recommend you to read that. But he basically described finding this white matter on his teeth and believing that there might be living animals in that. And so he takes some of this matter, he mixed this with rainwater in order to have prepared the samples, and then he used his basically a magnifying glass, which is now referred to as the van Leeuwenhoek microscope, and as he said, surprisingly found many small living animals which move themselves very strangely. And it's actually worth noting that van Leeuwenhoek was not only the first one to discover the microbiome, he was the first one to start cataloging the components of the microbiome. And he drew these beautiful drawings of all the different species that he found in such detail that we can actually guess which species he saw back then, right? He then goes on to do some nice comparative analysis, looking at the species that he found on his own teeth and comparing them to those that he found in kids and in women and in sober people and in drunk people and so on and so forth. And he concludes this letter with this really beautiful statement that this number of these animals on the scarf of Amentis are so many that I believe they exceed the number of men in the kingdom. And so let's think for a minute on what has changed in the last 350 years. And I think it's very clear that we've made some advancement in microbiome research. Specifically, rather than using the van Leeuwenhoek microscope, we're using a slightly more advanced, sophisticated, and also a bit more pricey machinery. Van Leeuwenhoek was able to characterize 5 or maybe 10 species, and we're able to characterize thousands of species and even more strains. And what's important, and I'll talk about a lot of that today, we're now able to also characterize other components of the microbiome beyond species, like genes and transcript and protein and metabolites. But if we think about the kind of analysis that we're doing, it is actually, I want to claim, not very different from what van Leeuwenhoek did. We still, in the majority of studies of microbiome, focusing on making lists of species and genes, making catalogs of the components of the microbiome, and comparing those lists across different individuals, across time, between different diseases, and trying to find variation in this list of components that is associated with either a disease of interest or between different data sets. Right? And I think it is also clear to all of us that this kind of van Leeuwenhoek kind analysis is limited. Just like if I want to understand what an iPhone is doing, or if I want to understand what is the difference between an iPhone and a Samsung Galaxy phone, it is not enough to just put it in a blender, which is exactly what we do in most microbiome studies, get all the components and look at the list of the components, say there are a few things here that are not here. And the reason it's limited, and this is the premise of system biology as a whole, is because in those kind of system, and specifically complex system like the microbiome, a lot of what we see has nothing to do with the list of components. It's all about interaction. It's all about how those different components relate to one another. It's all about the mechanism that are taking place in the microbiome. And by and large, this is a challenge that my lab is trying to tackle. We're trying to see how can we integrate method from system biology, from computational modeling to microbiome research in order to really move beyond cataloging and finding associations and really trying to get into a more mechanistic study of the microbiome. And we do that using several lines of work that I'll go on just by way of introduction very quickly. And so the first line of work is really just to try and build models of the microbiome, build a metabolic model that describes not one species, which is something that we know how to do relatively well, but how do we describe a model, a metabolic model of 2 species that interact, of 100 species, of 1,000 species, in a way that is both mechanistic and predictive, right? And if we can do that, we hopefully can understand how to predict potentially the microbiome response to diet, the microbiome response to different environmental conditions, how does the microbiome synthetic capacity changing with different conditions, and ultimately how can we design specific perturbation that will have a specific behavior that we are desiring. The second line of work has to do with using this kind of system biology approach or system thinking in general to analyze high throughput microbiome data and to integrate multi-omic data. And I'll talk about this in a minute. And just for completeness, I will say that the last line of work that we work on in the lab is basically applying those approaches to study many microbiome systems and see whether we can really infer additional observation about those systems. And in the talk today, I'm really going to focus almost entirely on this line of work in my lab, which is really concerning trying to develop new methods and new approaches to analyze microbiome data, and specifically to analyze multi-omic microbiome data. So again, just as a quick motivation, as you probably all know, in the microbiome world, we are now in the multi-omic era. And so if 10 years ago, when we had a microbiome sample, most of what we were trying to characterize and were able to characterize is a set of species in the sample using 16S, RNA assay, or many other technologies that have been developed. About 5 years ago, we've seen more and more studies that are trying to use shotgun metagenomic, basically taking the entire metagenome, lysing that, getting small bits and pieces of DNA, mapping them to gene catalogs, and getting a sense of what genes do we have in each sample. In many cases, we're not sure which gene belongs to which species, but we have a good understanding of the functional profile of the community. And then more recently, we're seeing additional studies that are, as I said, characterizing other aspects of the microbiome, including transcript and proteins and metabolites. And so I think it's also very clear to all of us that just characterizing those different facets of the microbiome for each sample and studying each of them independently is not sufficient. We do not want to be the 7 blind scientists that are trying to study an elephant, each getting into a totally different answer because they're looking at different facets or different aspects of this organism. But rather, we need to start to think about integrating those multi-omic data. And again, we've seen many of those studies coming up recently, really exciting studies. For example, we've seen studies that are trying to correlate the set of gene that is found in the metagenomes and the set of transcript that you see in a microbiome sample, and finding that overall there is a pretty high correlation between what genes are present in the microbiome and which genes are transcribed. We've seen multiple studies, really exciting studies, and I think we're going to see many, many more of them in the coming years that are characterizing both the composition of species in each microbiome sample and the composition of metabolites, basically using mass spec to characterize the concentration of different metabolites. And many of these studies then go on and try to identify associations between the abundance of each species and the concentration of metabolite as a way to generate hypotheses as to which species is responsible to variation of specific metabolite. But again, as I mentioned before, those studies are really focusing on statistical association, which is a great way to start query the data, a wonderful way to generate hypotheses. But to some extent, it is limited because it is actually ignoring much of the biological, ecological, and genomic data that we already have. For example, we can use our understanding of microbial ecology, organismal behavior to try to understand or to build a model a priori before analyzing the data on how the presence of different species in the community can affect the genes that each one of them will transcribe, right? We can use our understanding of metabolic activity and enzymatic activity in order to try to understand how the presence of different enzymes in the microbiome will affect the concentration of metabolites. And so on and so forth. There are different mechanistic understandings of different links between those omics that we can utilize, not in order to understand what the correlation that we have found tell us in retrospect, but rather as a forward inference approach, try to build an understanding of how two omics should relate to one another, and then use this understanding to analyze the data and hopefully get into a much more mechanistic type of analysis that is not only based on association. And this is one of the things that my lab is trying to do, is trying to see how we can integrate this kind of genomics, ecological, and biological data in order to build a model that links different omics to one another, and use this model as part of the analysis itself, not as something that I, in retrospect, look into after an association was identified. And in the talk today, I'll show you several such examples, and I'll focus mostly on integrated analysis of probably the two most common type of data that we now generate in the microbiome, which is species composition and gene composition, or if you will, the ecological profile of the community and the functional capacity profile of the community. And what I'll try to convince you is that what links the two is simply genomics. So let me just make sure that we're all on the same page in explaining what are these two profiles and why genomics is the linking factor between the two. And so think you have some microbiome sample, which is composed of a mix of different species at different relative abundances. What makes the species different is, of course, not their color, but the set of genes that each one encode, which are highlighted here as letters. And as I mentioned before, you can take one of two approaches, and we see many studies now that are doing exactly that. You can start by characterizing the set of species in your community, basically using 16S or MetaFlan tools or other tools in order to understand what is the relative abundance of each species, and you can generate those beautiful pie charts that will tell you that about 10% of your community is species A and 5% is species B, and so on and so forth, right? You can use, however, shotgun metagenomics to ignore the species themselves and just focus on the genes, and as I said before, map those reads into some gene database and characterize the abundance of each gene family in the microbiome, or each metabolic pathway, or each functional module in the microbiome, and again, generate those great plots that will tell you how much of the metagenome is involved in function A versus function B. Right? But I think it is clear to all of us that these two profiles of the microbiome, the taxonomic profile over here and the functional profile over here, and again, I'm going to use functional profile as a way to represent the set of genes in the microbiome, these two profiles are not independent. And what links the two is the genomic content of each of the species, because put simply, the metagenome is really nothing more than an aggregate of the genomic content of all the different species that I have in my community, weighted by the relative abundances, right? And I can represent it as a simple mathematical model that will tell me that the relative abundance of each gene in the metagenome is a linear combination of the prevalence or the copy number of this gene in each genome in the community multiplied by the relative abundance of these specific species. And there are a few normalization steps that need to be made, but roughly this is the correct model, right? And so what we have here is a very simple mathematical model that links taxonomic information, genomic information, and metagenomic functional information. Now, I want to assume that this model is in the back of the mind of everybody doing microbiome or metagenomic research. What I think is surprising is how underutilized is this model in analyzing this data, and we see multiple studies that are generating both taxonomic information and functional information and analyze these two omics in a completely independent manner. And what I'll try to show is that by taking this model into account, we can start to actually generate more in those scenarios where we have this kind of multi-omic data. So what can we do with this model? Well, the first thing is probably the most simple one that we can do is to visualize those links. So this is a snapshot from a browser that our lab is now developing. We hope to release it in about 3 or 4 months. It is called Burrito. And basically, it is characterizing, and we've seen a lot of those plots in microbiome research. We'll see many more, I guess, which is characterizing across multiple samples the different microbial clades that are present in each sample. This is another thing that we see often, which is the different functional pathways and their abundance in each microbiome samples. But what this browser is doing is putting them side by side and linking the two so that if, for example, you mouse over one of the species it will highlight which part, these are those dashed line, which part of the functional profile of the metagenome is being contributed by genes that are encoded in this specific species. And so you can easily look across those different profiles and understand why having more of these species is creating a profile, a functional profile that has those genes. And there are a few other features that I'll not mention. There are a few other things that you can do with this model. For example, and this is a project that we never intended to do, but one of the things that we realized that you can use this model to actually correct biases in metagenomic samples. And so one of the postdocs in the lab, Elhanan, when he just started working in the lab and annotating metagenomic data, was really looking for a way to see how successful he was in his annotation pipeline. And of course, as we all know, one of the challenges in microbiome research is that we don't really have good gold standard, right? How do you know that the functional profile, the set of genes that you found across different samples, really are representing what's in this sample? But he looked at this specific model and he made a really interesting observation, which is if he could identify a set of genes that are present in every genome that we know of microbial species, and always in one copy, and we call those universal single copy genes, and he looked for them and he found about 40 such genes that he could characterize. What he claimed is that the abundance of those genes across different metagenomic samples should actually not vary across samples. Because if each bug brings exactly one copy to the metagenome, then the specific taxonomic composition of the sample is not important, right? There should still be the exact same amount of those genes in each sample. And he thought that that could be a good benchmark to see whether those samples are really annotated correctly. However, when he actually started to looking at this collection of these genes, he found that this was not the case. So this is the abundance of those 40 genes across Human Microbiome project samples from different body sites. And as you can see, there is quite a bit of variation in how abundant those genes are in different samples, right? And so something here doesn't make sense. In fact, to make things worse, when you try to analyze data from multiple studies, and this is something we often do in our lab, and he specifically took samples from the Human Microbiome project, from the MetaHate project, and from a large study of type 2 diabetes, and he did a PCA plot to see how much variation there is in these genes, you see these beautiful clusters according to studies. However, these are all healthy individuals. The last thing that we want to see when we are trying to analyze data from multiple studies is clustering by study. Right? And so there is definitely something going on, and Ohad took a bit of time to look into that. And what we realized that there are several factors that are causing those spurious variation that should not be there. Again, those genes should not change across microbiome. The variation is caused by differences in the average genome size in every sample, so it has to do with the normalization that we tend to do in functional metagenomics. It has to do with our ability to detect the presence of various genes in different genomes, and so the closer the reference genome you have to the genome that you actually have in the sample, the easier it would be to actually identify them. It has to do with specifically some genes that are harder to identify than others because of their GC content, because of other elements. And without going into too many details, they basically develop a method that is called MUSICC. It's available on our website. You can actually download that, or you can run it online, that is using those universal single copy genes as a yardstick to normalize everything else and to try to solve this problem and to try to really figure out how your metagenome should be normalized and corrected. And just to go back to this plot that I showed over here, if you apply this method to those samples, as you can see, we cannot solve the problem completely, and there are a few things that are causing real differences between those studies, but at least now they looked a bit more similar than they did before. Okay, so this is a method that if you are using functional metagenomics analysis, you can use, and one of the nice benefits of that rather than representing gene abundances or functional abundances, relative abundances, which to me is really something that's hard to interpret, it is translating the abundances into average copy number. It basically will tell you what is the average copy number of each gene across genome in your microbiome sample. One of the things that we realized, by the way, is that those kind of genome-based regularities, those kind of dependencies that we see in metagenomic samples that are not biological in nature, but because of those genomic dependencies, are causing much more trouble than we thought. So I show you here an example of those kind of features making or bringing about spurious variation. It turned out that what's worse is that actually mask a tremendous amount of true variation that you have between different samples. I'm sure many of you have seen a picture like this, or from the Human Microbiome project, or from many other projects, and this is kind of one of the hallmark of microbiome research, that if you look across multiple samples, you see tremendous variation in the taxonomic composition of the community, but relatively high uniformity in the functional composition of the community. And part of that, by the way, is true, but one of the things that we discovered, and this is still in revision, is that this actually has to do with the way that we tend to process metagenomic sample without taking into account genomic properties of the different members. For example, even if you just apply MUSICC, which is a tool that I mentioned before, and you look across HMP stool samples at a specific pathway that actually makes sense to vary across individual, this is the kind of variation that you see when you don't have, when you're using the normal pipeline. If you apply MUSICC, you start to see a much stronger aspect of how much variation you see. And in this specific work, we developed 3 other features or 3 other factors that you need to revise the processing of the sample. And we started to see much more variation than we did before. For example, if you look at this specific pathway, even the standard metagenomic processing pipeline that everybody is using, when you compare between sample from health individual and sample from patient with type 2 diabetes, you see no difference whatsoever. Once you apply our revised pipeline, you're starting to uncover this hidden variation and start seeing additional pathways that actually are differentially abundant between health individual and type 2 diabetes. Right? And again, I'm hoping that this paper will be out soon, and there are several features that we argue there should be changed about the way that we process microbiome data. The other thing that we can do with this kind of model, which I think is worth noting, is actually looking for those instances where this link between genome and metagenome and species break down. Right? There are cases where what we expect to see in the metagenome based on what genomes we have and what their abundances are actually doesn't fit reality. And there could be various reasons for that. But the one that I want to note is strain level variation. So one of the things that is built into this model is the assumption that we know what is the genomic content. We know what is a set of genes that is encoded by each genome, by each species that we have in the microbiome. But of course, if one sample have a specific strain and another sample have another strain of the same species that differ in the presence or absence of specific genes, this metric will no longer hold. And so again, this is a study that was already published. I'll not talk too much about it. I'll just mention that Sharon Greenbloom, a grad student in the lab, was trying to identify those cases where when you look across samples, something doesn't match your expectation, and use that to actually characterize strain level variation across metagenomic samples. You develop a nice pipeline that allows you to do that directly from metagenomic samples. Just to give you the take-home message from this paper, when we actually applied it to sample from a large cohort, we find a widespread and adaptive strain level variation, not only in terms of single nucleotide polymorphism, but in the presence and absence of genes and in the copy number of genes. And what you see here are about 40 different species that we've analyzed. These are all their genes, and every bar here that has a color represent a gene that across about 100 samples that we analyzed has a tremendously high copy number variation. And as you can see, in some species, this is as the number of genes that have this feature. In some species, up to 25% of their genes actually differ in copy number or in present absence across these 100 samples. Right? And so when we said that you have a specific species in a sample, it actually doesn't tell us much in terms of what is the genomic content, given the fact that there is so much strain level variation. And so those examples that I showed before of different tools are basically a way to use this simple model that link taxonomy, genomes, and metagenome in a way that allows us to analyze those data better. The next story that I'm going to tell you is about a tool that is just now being developed. The paper is now in press. And I think it's going to be much more useful to many of the people in the room because it addresses, I think, one of the most fundamental questions that we all want to understand when we study the microbiome in a clinical setting, and that has to do with comparative analysis. And this project basically came about from talking to some of our collaborators and said, look, it's really nice that you can explain why there is a specific pathway in a specific abundance in a sample, but I'm interested in shifts in the microbiome, not just in their abundances. And so if you go back to this figure that I showed before about taxonomic profile and functional profile, in the clinical setting, in most cases what we have is a case where we have multiple samples. Some of them are labeled as cases, some of them are labeled as controls, be it a specific disease, a time point, whatever might be interesting. And as before, you apply taxonomic profiling. you get an estimate of the abundance of species in each sample, you create those bar charts, but what's really interesting to you in most cases is comparing the cases in control and identify specific taxa that are differentially abundant in your cases, right? And you can do a functional analysis, doing exactly the same approach as before, apply shotgun metagenomic, estimate the abundance of each gene or each metabolic pathway in your metagenome, create those estimates, and then compare them to identify specifically functions, either genes or module or pathway or any other kind of functional group that is more abundant, that is enriched in the cases. And what our collaborators wanted to know is, can you link these two? Basically, can you tell me why do I see a shift? Why do you see a difference in the abundance of some functional pathway in my sample? I don't care about one sample. I can't learn anything from that. I want to understand the shift that I see in a disease. And they want us specifically to address questions such as, if I'm seeing those kind of functional level shifts, does it have to do with one specific species that is changing in abundance and causing this increased abundance of a gene? Or is it always about community-wide dysbiosis? Is it always a mixture of different species that are affecting the functional capacity of the microbiome? And can you actually give us a number that will quantify exactly the contribution of each taxon in your microbiome to the enrichment that I see in each of the function in disease? And more generally, can you use this approach to identify intervention target, right? If I see a specific functional imbalance that I'm hoping to rescue, which species should I target? Which species should I add to the microbiome and try to reduce its abundance in order to reverse this specific shift? And in this project, when we first heard about this, and we thought, well, we understand the link between taxa and function, and we understand how genome contribute to that, and so that should be easy, and so give us a couple of weeks and we'll give you a great answer. It turned out that it was a bit more complicated than that, and it took us about 2 years to solve this, because what we didn't realize at the moment was that decomposing shifts is a much harder problem than decomposing abundances. In other words, understanding why you see a shift in a function, even though you have this specific model, even if you understand exactly where does each gene come from, right? So you've decomposed the composition of each sample, understanding why you see the shift is actually a much harder problem. First of all, because there is no trivial mapping from how much a species is contributing to the abundance of the function to how much the species is contributing to the shift in the function in cases. A simple example of that is a species that is highly abundant in both cases and control. It will contribute dramatically to the abundance of all the genes that it carries, but it will not contribute at all to the shift because that's not the species that's causing this shift between disease and control. Also, the shift metric is not linear. A lot of the studies that I showed before are relying on the fact that this simple model that we have is a linear model. It's a linear composition of all the different species, but once you're starting to think about shift, you're using t-statistic, you're using fold ratio, you're using Wilcoxon analysis to find things that are statistically significant between different samples, those metrics are not linear, and so a lot of the assumptions that we use to make actually tend to fall down. Another problem is that community contexts matter. The species contribution to functional shift is not dependent only on the changes in the abundance of these species, but also how does it co-vary in relation to other species. And you can think again about a simple scenario where you have two species that are negatively correlated to one another, and so they compensate for the functional capacity, and as a pair they actually do not contribute at all to the functional capacity shift that I see, but each of them might contribute in some way. And more conceptually, it turned out that mathematically, this is stuck for some reason. Doesn't work. That doesn't work either. Oh, these? Okay. Okay, perfect. Yeah. And so more conceptually, another problem that we realize is present is that, oh, thank you, is that the idea of decomposing shift mathematically was not existent. In other words, it was hard to even realize how do we define a contribution to a p-value. How do you tell how much of the significant signal that you see come from species A versus species B? And so Ohad, this postdoc that I mentioned before, had worked on that quite extensively for 2 years, as I mentioned, and came up with a really beautiful framework that is called fish tacos for a reason that I cannot, or rather not disclose. Basically, the framework works as follows. So it gathers input profile of the taxonomy of those different samples. So basically, the species composition across your both cases and controlled samples. Your metagenome-based functional profile. So again, an estimate of the abundances of different pathways in each of the samples. If you have genomic data, if you have reference genome of the different species in the community, we can use that as input. If not, we can actually infer this automatically from the set of samples that we have using a method called metagenomic deconvolution that we developed a couple of years ago. And then using some permutation-based analysis to quantify how changes in the abundance of each species contribute to the shift in the species. We use a method from game theory that's called Shapley value analysis. It was developed originally to try to decompose the contribution of players in a coalition in order to understand what does each species contribute by itself. And at the end of this entire process, the plot is basically a precise characterization of the contribution of each species to the shift in each function. And I know it's a bit hard to figure out what exactly is this tool doing, so let me give you a flavor of the kind of result that you can get with FishTaco. And so this is, for example, from analysis that we did, again, of a large-scale cohort that comparing healthy individual to individual with type 2 diabetes. And if you look at the metagenomes of this individual and you estimate the abundance of each genes, you find 3 different sugar transport that are significantly more abundant in patients with type 2 diabetes. And here I'm showing simply a functional shift score that is measuring some enrichment statistic. In this case, the W statistic that is used by the Wilcoxon test. And if this was the only analysis that you would do, the only thing you can say is that those 3 sugar transport are all enriching the disease, roughly the same level of enrichment, right? If you also have functional data, if you also have taxonomic data on these samples, you can also identify specific species that are more enriched in type 2 diabetes or are less abundant in type 2 diabetes, but there is really no way to link the species shift to the functional shift. If you apply FishTaco, what you'll see is that, right? So basically, FishTaco is taking the enrichment of the function and break it down into its specific taxonomic driver, and it will tell you that about 80% of the shift is being contributed by Firmicutes and about 15% by Escherichia, right? You may notice that the original enrichment score was over here, and the sum of all the small contributions that I have from the different species is actually not reaching the entire shift. And that is because one of the things that we realized when working on this framework is that there are actually multiple ways in which taxonomic dysbiosis can contribute to functional shift. All those species on the top are species that encode this specific sugar transport, are more abundant in type 2 diabetes patients, and as a result, create an enrichment of this function in those metagenomes. However, there is another group of species, including, for example, this Prevotella species, that is less abundant in type 2 diabetes patient and lack the genes that encode the specific sugar transport. And because we're looking at relative abundances, combined, they too contribute to the enrichment of this function. And there are also a few other group of species that actually push the enrichment in the other direction. As you can see, they're trying to reduce the level of enrichment, but they're simply not strong enough to overcome the other species that are pushing the function enrichment in this direction. So this provides us a precise characterization of all the different taxonomic contributor of this functional shift or functional imbalance that I see, and exactly what their contribution are. What was more exciting to us is that when we apply the same approach to understand the enrichment in the other sugar transport, we find a completely different pattern. So the second sugar transport is driven mostly by Escherichia. The third one is mostly by Bifidobacterium and a few other species. And that, of course, has quite profound implication for translational research, because if we want to start thinking about intervention targets, which species should we change their abundance in order to reverse a specific functional imbalance, it tells us there is a great deal of specificity in the link between the ecology of the microbiome and the functional shift. And so if I want, for some reason, for example, to reverse this functional shift, potentially without affecting the functional state of the other, pathways, there is one specific species I can try to target and be very specific in its impact on the community. It turned out that this pattern of specificity in the link between ecology and function goes beyond just different function, different paths. We analyzed many, many different data sets that were available to us. We see the same pattern if you compare across different anatomical sites in the human body. But maybe what was most interesting is when we compare across different diseases and looking at the same function. So this is, again, a FishTaco plot that you get when you're analyzing the enrichment of the methane metabolism pathway. If you look at this specific pathway and you look at the gene, it turns out that both patients with type 2 diabetes and patients with inflammatory bowel disease have an enrichment of this specific pathway in their metagenome. However, if you look at what are the taxonomic contributors of this specific pathway, you again find a totally different set of players. And so an intervention in order to rescue this specific pathway in one disease, of course, would be totally ineffective in the other. Right? And so again, we see that if you analyze these two types of data independently, you simply cannot see those specificities in the link between ecology and function. I would just very briefly mention that there are many other results that we included in this specific paper. For example, we show that if you use genome content inference, as you remember I mentioned that if you don't have a reference genome for your community member, we can infer them from the data itself. It turned out that that actually improved our prediction power and improved the agreement between the ecology and the metagenome. We show using a simulation study at this point that our prediction of contribution score actually matched nicely the effect that we think eliminating a species or adding a species the community might have, and so on and so forth. The most important thing to note is that Fish Taco is available online. It has been available for a while now. You can use it. You can download the tool. You can run it on your own data. We'll be happy to help with that. We also provide a web application that will generate the same Fish Taco plot that I showed before, automatically giving the data that you have. And again, we're happy to collaborate with any lab that is interested in using that. We already have quite a few labs that are using this tool in order to really understand the link between the ecology of the system that they're studying and the functional capacity of the system. And so everything I show you so far really has to do with linking genomes and metagenome, or the microbiome ecology with the microbiome functional capacity. I want to note that this is not the only two omics that we can analyze using a model-based approach. However, we'll need different models, right? And I want to very quickly just describe this project that was carried out by Cecilia Knocker, a grad student in the lab, that was taking on a really ambitious project. She wanted to ask, can I actually link the species in the microbiome, the ecology of the microbiome, not just to the genes that are present in the metagenome, but all the way to the metabolome of the community. In other words, can I explain the variation that I see in the metabolites that are found in microbiome samples across different individuals based purely on which species are present and what is the relative abundance in different samples? And our idea was, again, not to use statistical association, but to build a model of how these two omics should relate to one another. And so she said, look, I can use the genome, the species that I have, plus genomic information to build a model of what are the gene abundances in my metagenome, as I did before. We can use enzymatic data to understand what is the reaction capacity, what is the synthetic capacity and degradation capacity of each enzyme that is being encoded by the metagenome. And then we can use metabolic modeling and use a very simple modeling approach at this point to try to understand how the abundance of different enzymes in the metagenome may affect the concentration of different metabolites in the sample. Okay? And so again, this is a model, in this case, a combination of a mathematical model and a metabolic model that is linking taxonomic information, community ecology, with metabolomic information, which is, if you will, the ultimate phenotype of the microbiome. Right? Cecilia went on and built a framework that is allowing us to do that. It is called MIMOSA. Again, it's already available and you can use that. That is trying to do that. And I have to say that when we started to think about this project, we were very skeptic that it would ever work. And the reason for that is because in this link from ecology to metabolite, we're making significant simplifications about how the microbiome works. We're ignoring regulation. We're ignoring fluxes. We're ignoring gene transcriptions. We're ignoring the effect of the host in some cases, and I'll talk about this in a minute. We're ignoring the way that different species impact the activity of other species, and really just trying to look at how much of an enzyme is there and how much it can synthesize each metabolite and how much it can degrade other metabolites. But it turned out that when we apply this kind of framework to real data from the vaginal microbiome, from the David Frederick's lab, and we ask for each metabolite, can our model explain the variation that we see in the abundance of this metabolite across different samples, right, based on this model and the variation that we see in the ecology of those samples? It turned out that for about a third of the metabolite, this model by itself was sufficient to explain the observed variation. Right? And so again, in some sense, it tells us that just by looking at how the ecology changes the functional potential of the microbiome, the metabolic potential to synthesize and degrade different metabolites, we can actually predict quite nicely the the concentration of different metabolite, even though we ignore everything else. We ignore the host, we ignore the regulation, we ignore other factors, right? And the nice thing about this model, which is not necessarily a feature that you have in a statistical model, is that because this is a fully mechanistic, detailed model, we can then dive into the model of each of those metabolites that we have predicted correctly and ask which species really contributed to the variation that I see in this metabolite, right? And I would note that the results that we have found are actually different than the one that you see just by looking at statistical association, right? So in some sense, looking at the mechanisms that are involved would allow you really to understand those kind of contribution. And since then, by the way, we did apply this to gut sample. We see relatively similar results, both from David Frederick Lab and from Jenna Jensen Lab. And so I want to end with just saying that this is an interesting approach, but there are obviously many challenges ahead, right? So I noted that I showed you several examples. We're trying to link genomic, using genomic species information to functional information, using species information to metabolite concentration. But many of the other processes that are involved in the link between the different omics are not as well understood. There is definitely a lot that we can do, and we have some preliminary work on that, but it's not easy to understand how to build these models. What I think is more important is that there are several large-scale factors that are not clear as to how we should treat them. For example, what resolution should this model be using? How well-defined each model should be in order to be effective? How do we introduce the effect of the environment to introduce model? What scale do we need to build the model and characterize the microbiome? If you're looking at a temporal model, for example, do we need to characterize the microbiome over days, over the weeks, over months, over years? Another big problem is what we call in the lab the functional gray matter. So everybody in the microbiome field is talking about the functional dark matter, which are the species that we do not sequence. I have to say that personally, I'm much more concerned by the species that we did sequence, and we have their full genomes, but we don't have any functional annotation for about half of their genes, right? So we have the genome, but it really doesn't help us at all in order to build a mechanistic understanding of the microbiome. And so just to quickly summarize what I've tried to show you today and the take-home message, I do believe that applying this kind of system-based and model-based approach not only is a way to represent or to simulate the microbiome, but is a way to analyze microbiome data, to introduce those kind of understanding, those kind of ecological and genomic dependencies that we are aware of. As part of the analysis pipeline is crucial for really obtaining a mechanistic understanding of the microbiome. There are many tools available that are starting to do that, both from our labs and from other labs. And lastly, because I'm talking to, I'm assuming quite a few clinicians and experimental biologists, and whenever I talk about model, there is always, I see this question at the back of everybody's mind, but about this model is not really accurate, right? We're ignoring a lot of things. And so in order to address that, I like to quote people that are much smarter than me. The first one is Arturo Rosenbluth, that is a philosopher of science, that said that the best model for a cat is another cat, preferably the same cat. It should be emphasized that he said that in irony. You should not use the same cat to model your own cat, because then you cannot learn anything from the model system. A model system should be similar to the original system. But probably the best statement about this is by George Box, a well-known statistician, that said that all models are wrong, but some are useful. And so we know that our model is wrong. What I hope I was able to convince you today is that using this model is still can be very useful in order to understand the data that we are generating about the microbiome. And I'll end with that. I'll just thank all the people that did the work. I think I mentioned all of them by name throughout the talk, so I'll not do that again. I'm going to thank the funding agencies and, of course, our collaborator for providing some of the data that we analyzed, and all of you for listening to me at this early time in the morning in Seattle. Thank you.